This is iGEM. We are a student-based organization that focuses on solving global problems using genetic engineering. iGEM focuses on developing scientific knowledge, engaging the public, and collaborating within the community. Our first project uses two recently discovered proteins to change A-type blood to O-type, the universal donor. The first protein cleaves the NAG tag, producing an intermediate sugar and acetate. The second protein cleaves the intermediate sugar and leaves the O antigen. Our second project uses tissue plasminogen activator, also known as TPA, which is a protein already used to treat blood clots. By functionalizing microwheels, we can provide high local concentrations of TPA to increase effectiveness and reduce the potential side effects of prolonged TPA exposure.
Dante Disharoon, along with Dr. Mar's lab, have been working on microwheel control for blood clot lysis. Currently, Mar's group works with commercially available biotinylated TPA. The binding method currently used is nonspecific, leading to inactive TPA binding to the microwheels. Our goal is to use site-specific binding to improve the functionality of TPA. In the video, we can see the effect of TPA functionalized wheels compared to dissolved TPA, as well as the effect of different microwheel movements. Peter Raffeld and associates at the University of British Columbia have screened gut bacteria and found two enzymes, PODTR4 and PODTR5, that can convert A-type blood to O-type. We hope to use dynabeads functionalized with these proteins in order to develop a more physically realized blood conversion system. Our first goal is to perform topo-TA cloning on our designed inserts, and then subclone those inserts into expression vectors. Next, we will transform our vectors into the expression systems, E. coli for the blood conversion enzyme pair, and yeast for TPA expression. From here, we'll test and optimize the expression of each protein before moving on to test each system. For experimentation, we will verify the activity of each protein. The effectiveness of TPA can be tested using fibrin plates, where the area of degradation over a set time interval can be compared to commercially available TPA. For the blood conversion enzyme pair, we can measure the concentration of the byproducts formed, acetate and D-galactosamine. Once we have verified the activity of TPA, we can begin to perform experiments with functionalized beads and synthetic clots. For our blood enzyme pair, we'd begin testing the effects of co-functionalizing beads on the red blood cell conversion rate.